if you've heard my chatter, this is Adventures in Sensing. It's great to be able to take you guys on an intellectual voyage through all different kinds of really interesting and diverse and often extreme applications of sensor technology. We're living in the age of the sensor, and just incredible how you can get information from lots of things and what you can do with it. So this one, we're going to focus more or less on environment. We've got two really great, well-known practitioners here to talk to us about very different aspects of environmental sensing. And I'll let Carolyn introduce the speakers and take over from here.
Great. Thank you, Joe, and thank you to Professor Epstein and Professor Cocroft for being here with us today. Yeah, so just as Joe mentioned, we are going to be recording the session, so just so everyone's aware of that, it looks like the recording is on, so we're good to go. And in terms of format, each of our guests will give about a 30-minute talk, and then we'll have some time for some specific questions and answers related to that talk, and then we'll go on to the second one and do question and answer after that one. Yeah, so as Joe mentioned, environmental sensing is a very broad field that covers many different scales, regions, modalities, and I think we have a real treat here today. We'll start with, something a little more zoomed out, talking about remote sensing and satellite imagery techniques, and then zoom way in to look at insect communication. So our first talk today will be from Professor Howard Epstein at the University of Virginia, and I'll give a brief bio intro and then turn it over to Professor Epstein. So Professor Epstein is the professor and chair of the Department of Environmental Sciences at the University of Virginia, and his research specializes in the ecology of Arctic tundra, dry grasslands and shrublands, and temperate forests. His research focus is on vegetation dynamics, nutrient cycling, and plant-soil atmosphere interactions. He's published over 160 peer-reviewed journal articles and book chapters, led the annual production of the Tundra Greenness Essay for the NOAA Arctic Report Card, and has contributed to several international reports, including the 2019 IPCC Special Report on the Ocean and the Cryosphere. Let's see, he served on the board of directors for the Arctic Research Consortium and is the co-lead of the Vegetation Dynamics Working Group for the NASA Arctic Boreal Vulnerability Experiment. And he recently received a large grant from the NSF's Navigating the New Arctic program to study the interactions between the built and natural environments in the community of, I'm not going to pronounce this correctly, but Utqiagvik, Alaska, and I think we'll hear about that today. And just as one note, I'd ask people to mute themselves so that we can give our full attention to the speaker. So yeah, without further ado, I'll turn it over to you, Professor Epstein. The floor is yours. Thank you.
Yeah, thank you, Carolyn. That was a beautiful introduction. I appreciate that. And thank you for having me. I'm hoping that what I have to present to you today is useful or at least interesting to you. All right, share my screen here. Are you getting the full screen now?
Yeah, that looks great. You just
okay.
So perfect.
Great. I'm gonna get myself maybe a pointer here. Come on. Oh, didn't want to do that yet. Oh, there we go. Annotate. All right, I got myself a spotlight here if I need. All right, terrific. So yeah, so again, thanks for having me. What I'm gonna do, so I'm gonna talk about the new project, which is in Utqiagvik, Alaska. I think you pronounced it reasonably well. I'm not sure I pronounced it correctly. It's formerly Barrow, Alaska. So it's the northernmost city in the United States. But that's a brand new project. We don't have a lot going on the ground yet. So I decided to give you sort of three case studies, vignettes, I guess, with regard to environmental sensing. So we'll cover satellites, drones, and then the field instrumentation part will be the Utqiagvik project. And we'll start with satellites and then go down to the field. So, all right, starting with satellites, you may know or be aware that that we have numerous Earth observing satellites out there in space. These satellites take a variety of different shapes and forms and sizes and do a variety of different things. We have Earth orbiting satellites. We have geostationary satellites. The sensors on these satellites collect information at varying resolutions, so different spatial resolutions, different time frequencies, and different spectral resolutions or different resolutions of the information that they're obtaining. Some of these sensors are passive and they're just collecting information that is reflected or emitted from the Earth's surface. Some of the sensors are active, meaning they are sending something. So they may be sending a laser or radar signal that is then getting bounced off of a surface and then coming back to the sensor for recording. There are sensors that cover varying wavelengths, okay? So different types of wavelengths. There are optical sensors, there are radar sensors and others. Some of these sensors or some of these satellites and sensor platforms are put in space by federal government, by different agencies like NOAA and NASA, international agencies like the European Space Agency. And some of them are commercial, all collecting different types of information on the Earth's surface. So I'm a vegetation person, and I've largely worked with passive optical sensors. Some of the big ones that I've worked with are the Landsat series of sensors that basically go back. Those go back to the mid-70s. Pretty nice data set. And then the Aqua and Terra satellites have the sensor called MODIS on them, and those have been around since about 2000. So those are the ones that I've used most frequently, and some other satellites as well. So let me tell you why this is useful for someone who's studying vegetation. So this is you have wavelengths on the wavelengths on the x-axis, and you have reflectance of those different wavelengths on the y-axis. And the bands that are shown here, these gray bars here, those are bands from a sensor that was on on certain Landsat satellites. So these are the Landsat thematic mapper bands. And you can see here that from a vegetation perspective, the big difference in the spectral signature of vegetation and the spectral signature of bare soil occurs in largely two areas of the spectrum. So one here you can see my pointer, right?
Yeah, we can see it.
Cool. So right here you can see that the vegetation is reflecting a lot less in the red wavelengths than the bare soil, and this is because of photosynthesis. Chlorophyll absorbs red light for photosynthesis. And so vegetation is absorbing more of the red light, reflecting less of the red light. And then the other big difference is in the near infrared. Vegetation has a lot of near infrared radiation, as opposed to the bare soil, which absorbs more of the near infrared radiation. So you get these big differences. But just to orient you, this is, so the thematic mapper bands, this would be blue and green and red, and these are 3 near infrared bands, and then there's also a thermal band that's out in the longer wavelengths. So there are ways of distinguishing vegetation from bare soil using the near infrared and the red wavelengths. And you can derive an infinite number of indices from the information that you collect from these sensors. Probably one of the most common indices that we use for vegetation is called the normalized difference vegetation index. And it's based on what I just showed you in the prior figure. So it looks at the difference between the near infrared reflectance and the red reflectance. And then that's the difference part of the term. And then it divides by the sum of the near infrared and the red, and that's the normalized part. So it's the normalized difference reflection, normalized difference vegetation index. And well, you can see that going back here, the difference between the near infrared and the red, right, will tell you a lot about how much vegetation you have. And so this is just an example here. We've got green vegetation. It's reflecting high in the near infrared. It's reflecting very low in the red, and your NDVI is 0.72. If you've got sort of browning vegetation here, it is reflecting somewhat less in the near infrared, but reflecting quite a bit more in the red because it's not photosynthesizing necessarily, and you get an NDVI of 0.14. So you can see the difference. The utility of this index and the other indices is pretty amazing. I mean, you can do a lot with this information. And here are some other ways of capturing the NDVI and utilizing that index. So this is the seasonal dynamics of the NDVI. And this is just an example using that using the MODIS data from the Terra and Aqua satellites that I showed you. And this is just looking at 2 time periods, 2001 to 2008 and 2009 to 2018, to see what happened between those 2 time periods. But if we look at the seasonal dynamics, so before the growing season starts, the NDVI is close to 0 or less than 0. Once the growing season starts, the NDVI kicks in. It reaches a peak at some point in the growing season, and then as the vegetation starts to senesce, the NDVI decreases. So there are some things you can pull out of the seasonal curve. You can pull out the maximum value of the NDVI, which is something that we often do. And you can calculate the integral under the curve, or the time, the TI, the time integrated NDVI. And that's an index of the total productivity of the vegetation over the course of a growing season. The maximum is the peak biomass of the vegetation. And well, we can look at how these things changed over time, which is commonly what we do. And so we've used these 2 pieces of information, the maximum NDVI and the time integrated NDVI, to look at what's going on with Arctic vegetation. And we have been involved with, as Carolyn said, writing the tundra greenness essay for the NOAA Arctic report card every year. And my former graduate student, whose name is interestingly JJ Frost, working in the Arctic, and he took over the lead of this essay on the Arctic report card. And basically, what we're seeing in the Arctic is the Arctic is greening. This is the maximum NDVI over, and this is actually a 38-year record. This is a different. This is a sensor on NOAA satellites. The sensor is called AVHRR, which is advanced very high resolution satellite, advanced very high resolution radiometer, which kind of ironically, it's not very high resolution, but at the time it probably was when it first started in the early 80s. But basically what we're seeing is the tundra is greening, and you can see that this is a projection from the North Pole downward. And you can see that there are some areas that are where the NDVI is decreasing, particularly in the Canadian high Arctic here, in this area of Western Alaska. But the general picture is that with climate warming, the tundra is getting greener. And we are using this satellite information to analyze the dynamics of tundra greenness over this almost 40-year record. Okay, so this is actually, even though the name of the sensor says very high resolution, it's pretty coarse. It's sort of the data range from 1 kilometer to 12 kilometer resolution, which is actually not very high resolution at all. So we want to get it. We wanted to get at some of the mechanisms, like what's causing the greenness. And so we looked for higher resolution information. And one of the and we wanted to go back. We wanted to see if we can go back even further in time. One of the cool things that we took advantage of is that in the during the Cold War, the U.S. was and other countries were doing quite a bit of spying on each other. And so we took advantage of a spy satellite program that the U.S. had implemented in the 1960s. And this was following the U-2 mission, the U-2 spy missions. And so the first satellite, the first spy satellites were put into space in the early 1960s, and we started to collect some good data from them in the mid to late 60s, and that extended through the 70s as well. And these data became declassified during the Bill Clinton administration. And so a bunch of the data were declassified in 1995, and the remainder of the data were declassified in 2001. And these are really cool. So what the satellites could stay in orbit for, not for very long. I think they were able to stay in orbit for just a couple of weeks. And they would take photographs of the Earth's surface and then eject the film, and the film would be connected to a parachute, and they would float down, and these planes would go along and scoop these things up in midair and bring them back for analysis. And so it was pretty nice utility for environmental sciences. I'm sure it was for spying as well, but it became very useful for land surface change detection. And the resolution was pretty high. The ranges, depending on the camera, range from 0.7 to 2 meters, depending on the camera and the height of the sensor. And so this is an image from one of those missions called the Corona set of missions. This is 1968. This is a tree line, shrub tundra tree line site near Harp, Northwest Siberia. So this is just north of the Ural Mountains. And you can see that it's pretty nice resolution. And all of the darker areas are these guys. So the darker areas are these alder shrubs that are basically expanding their way across the landscape because of a variety of factors, including air temperature warming and disturbances. And so here's the best time series or paired set of images that we have. This is from a similar mission called Gambit, 1966 image, also in Siberia on the Western Taimyr Peninsula. And we paired this with a high resolution commercial satellite image from GeoEye. And I'll just flip back and forth, and you can see that there are big differences in the shrubs between those two time periods. So this is some pretty dramatic shrub expansion over a 40-year period. So this tall shrub expansion is one of the mechanisms that's causing the greening of the Arctic. And just to put it into a broader context, we did this kind of analysis, this paired image analysis using the spy satellite imagery as our time 1 and commercial imagery as our time 2. And we were able to identify a rather ubiquitous increase in shrub cover, in tall shrub cover, particularly that one that I pointed out, which is Siberian alder, over a 40-year period in the Siberian tundra. So that was really neat. And that is the end of my first case study on satellites. And now I'm going to move to Virginia, and switched to some drone technology that we're utilizing. And this is the title of this study is to assessing the ability to detect invasive plant species using drone-based leaf scale, visible, and near-infrared imaging spectroscopy. This project is largely being done by a PhD student of mine. Her name is Kelsey Hulsman. And one of the targets, so she's looking at a variety of invasive species, but we're going to focus on one for this particular case study. And one of the target species she's looking at is autumn olive. It's an exotic from Eastern Asia. And you can see that it has spread pretty widely throughout the northeastern United States and the Midwest. Here's some photos of autumn olive. So Kelsey's overarching question, at least from the autumn olive perspective, is how do we detect invasive species? So this would be a really amazing tool for conservation practitioners. Okay, if we can develop algorithms using spectral signatures, if we can develop algorithms for identifying individual invasive plants, we can then fly drones or maybe other platforms with other sensors over large areas, identify invasive plants, and then use mitigation strategies. And this is opposed to just going out into the field and hiking around the field and finding where these species are. So this would be a real bonus for conservation efforts. So can we do this? Can we detect individual plants and individual plant species using these technologies? So a more specific questions is, can we derive a spectral signature from individual pixels that can be used to detect target invasive species? On the flip side, is there too much intra-individual variability or intraspecific variability? So variability in spectral signatures within a species to be able to identify that species from and differentiate it from other species. So that's the goal of this. Okay, this is our platform. Our drone is the DJI Matrice 600 Pro. It's a hexacopter. And the payload on that is this hyperspectral camera from Headwall. This is a company, Headwall. It's a Headwall Nano. And it is a visible near infrared sensor. This particular one goes from 400 to 1,000 nanometers. I would love to go even further, out to 2,500 nanometers, which gets you a lot of information in the shortwave infrared, but that is heavier and more expensive and it's just not feasible for us at this point. This records 270 spectral bands. So the Landsat thematic mapper gave us 7. Okay, this is giving us 270. So the bandwidth is 2 to 3 nanometers. So every 2 to 3 nanometers, this is collecting information. And it's very high spatial resolution too. We can get 3 centimeter by 3 centimeter pixels, depending on the height and the speed of the flight. And so Kelsey would collect training data. She would collect 2 sets of data, one to train the data set, develop the algorithms, and the other to test and use the algorithms and see whether they worked. And so she collected imagery many, many times over the course of the 2020 year and growing season. But we're going to focus on April and June 2020, looking at autumn olive. And here are some examples. Here's some data, actually, some data of different species and their spectral signatures. So here, this is you can't read that, but know that each of these colors represents a different species. And this should look like that vegetation spectral signature that I showed you earlier There's high reflectance in the green, right? Because vegetation is green, so it's reflecting very highly in green wavelengths. It's photosynthesizing in the red. Actually, it's photosynthesizing in the blue too. So it's high absorption in the blue, reflectance in the green, absorption in the red. Then you get this area called the red edge, where the reflectances go way up. And then you've got this near infrared plateau here. And you can see the species there's some differences there among the different species. This is April, when the species are sort of just kicking in. And this is June, where we're at maybe getting into the peak of the growing season in central Virginia. We're zooming in now on the blue, green, and the red. So here's the blue, the green reflectance, getting into the red absorption here. And from an autumn olive perspective, it really distinguishes itself in the blue wavelengths during April, which is a very nice feature and something that would be super useful to be able to pull out the spectral signature of autumn olive and identify that as being different from other species on the landscape. So Kelsey honed in on a few sets of wavelengths, knowing that she has 270 pieces of data for every pixel. Some of the wavelengths that seem to do the best job of discriminating among species were in the blue wavelengths, the red wavelengths, and the near infrared wavelengths. Not surprisingly, these are the blue and the red are pigment related, and the near infrared is very much, near infrared reflectance is very much driven by vegetation. And so she ran a partially square discriminatory analysis for both April and June for autumn olive versus everything else, and it worked out pretty well. So this is the multivariate space, component 1 and component 2 of the multivariate analysis. And the green pixels are all data from autumn olive, and the gray pixels are all data from anything that's not autumn olive. And you can see the spectral signatures. So their spectral signature in the blue, red, and near infrared separate these two out reasonably well in April and also reasonably well in June. And so she was able to develop algorithms to determine what is to make predictions about what is an autumn olive and what is not an autumn olive. And then she tested those algorithms on her test data set, and it turned out rather well. So in April, we had 2 false positives. So there were two individuals that we identified as autumn olive that were not autumn olive. And in June, there was there were 2 false negatives in April, meaning we said that we identified 2 species as not being autumn olive, and they were autumn olive. And then 1 false positive in June, where we identified a species as not being autumn olive, and it was autumn olive. So reasonably well. We had most of what we found were true positives and true negatives. All right, moving on. Okay, this is the last case study, and I think I'm doing reasonably well with time. I'll try to wrap it up pretty quickly so we can have some time for questions. So the title of this project is Understanding the Changing Natural Built Landscape in an Arctic Community and Integrated Sensor Network. And we're going to move now from sort of space-based or airborne based to ground based measurements. This is our team. We have a really interdisciplinary team. We have architects, we have environmental scientists, we have a social scientist, we have a data scientist, and we have collaborators from the Army Cold Regions Research and Engineering Laboratory, from the also government agency Cold Climate Housing Research Center, and different local indigenous organizations, and some new graduate students on the project. So we have three main research components. We want to understand how urban systems interact with the surrounding air, ground, and water conditions. From an architectural perspective, we want to generate environmental design analyses and design guidelines for current and future management of the built environment in Utqiagvik. And then we also want to produce this knowledge with local residents, and we want to study how we produce the knowledge with local residents. So how effective are we at collaborating to generate this information? Looking around the city of Utqiagvik, there are some areas that we may focus on. So here, so I'll start. Number one is sort of the built lagoon interface, built environment lagoon interface here. Two is the built environment coastal interface here. Three is the built environment tundra, maybe natural tundra interface here. And then more of a lagoonal system in number four. The network that I'm most involved with, and that I'm sort of presenting to you here, is our terrestrial sensor network. So we're going to put out an array of terrestrial meteorological sensors within the city of Utqiagvik, and we're actually installing them. And because of COVID, we haven't been able to get out of the field. So we're installing them on our UVA property in Charlottesville to test the equipment and all the logistics and all the data issues. And it's going rather well. We have a beautiful setup on a gorgeous property that the University of Virginia owns. And I'll just show you some of we went out there the other day. Here's some of the photos. So here are some of the things that we're collecting. So we have wind direction here. You have wind speed up right there, wind speed right there. That's a pyranometer, so that's going to give us incoming solar radiation. This is housed in here are air temperature and relative humidity. This is a precipitation tipping bucket gauge, rain gauge. And what you can't see are the ones in the ground, which are actually for the Arctic. Those are the most important ones. The ground temperature and ground moisture are probably the most important variables that we're going to collect. And one of our main goals is to understand how the built environment interacts with climate change to affect heat fluxes going into the ground, changing ground temperature. The reason we care so much about changing ground temperature is because this city is underlain by permanently frozen ground. And if that ground thaws or large chunks of ice in that ground melt, then you'll get slumping of the land surface and collapsing of buildings. So that's a variable that we're super interested in. We're also not seen here. We're also interested in snow depth and accumulation, and also potentially some air pollution or constituents of the lower atmosphere. Again, similar setup here. We've got a wind speed and direction up here, and then we've got a wind speed and direction down here. So we are one ultimate goal maybe to get some information at different heights. And you can see that we're putting some out in the open. We're putting some right on buildings here. And you go out there and they're doing different things. At the same time, you watch the sensors that are right near a building, and they're doing very, very different things than ones that are out in the open. And this could mean very interesting things for what's going on in the ground, both from a moisture and a temperature perspective. All right, I'm going to try to wrap it up quickly, but just to let you know, we're also doing aquatic sensing. My postdoc, Claire Griffin, is leading this effort. Things we're looking at in the water bodies are conductivity, temperature, pH, dissolved oxygen, dissolved organic matter, turbidity, which is suspended, the degree of suspended particles. And we're going to do this in lagoons and ponds and just a variety of water bodies throughout Utqiagvik. We're also doing some geotechnical surveys. This is what the Cold Regions Research and Engineering Laboratory is providing for us with our collaboration. They have sensors that will collect really important information for us, such as the active layer. The active layer is the depth that thaws in the summer. So we want to see, is the active layer changing? Is the active layer getting deeper as the air and ground temperatures? So they can run transects and get really nice data on the depth to the active layer. The other thing, they have ground penetrating radar, and the other really important piece of information that they can get for us are ground ice concentrations. So if you take, like, so permafrost is frozen whatever. It can be frozen rock, it can be frozen soil, or it can be frozen water, it can be ice. If you take frozen rock and soil and you go from -1 degree to +1 degree, it's no big deal. But if you take obviously ice and go from -1 degree to +1 degree, it is a big deal because you change the phase of water from frozen to liquid, and that changes the density, and that makes the ground slump. So they use ground penetrating radar to show us what the ice content of the ground looks like, so we can really get a sense for the vulnerable areas on the landscape. So those are the main sensing things that we're doing. We have, and I'm going to stop. We're going to be generating lots of data. So we have a big data governance and management effort. And we also are very much focused on this collaborative production of knowledge, where from the very beginning of the project, we're working with the community in Utqiagvik to determine where the sensors are going to be, what extra things do we want to measure, and how basically we're going to go about conducting this research. So that is all. I'll wrap it up. I hope that was somewhat useful, informative for you. And if there is time, I'll take some questions.
Fantastic. Thank you so much. That was a great talk. I loved the 3 different scales. Yeah, I think we have about 5 minutes for questions. So if people want to, I think we're, a good sized group if you just want to speak out, or you can put a question in the chat and I can read it out.
Maybe a quick question while we get organized. Lots of questions. Great talk, Howie. Lots of questions. We could talk for an hour, and hopefully we will soon. But you have to power this thing through the Arctic winter. Is that why you have that big solar cell? Because most of the sensors probably are pretty low powered. You do second one down, but that, how are you going to keep this thing going during the winter when there's very little, if any, light up there?
Yeah, we're going to use solar. We're going to use solar to keep them going during the winter, and solar powering batteries, and the equipment that we have installed locally. They will those batteries will go for they'll go for almost a year. So if they can power fully during the summer, they can last through. They should be able to last through the winter. We're still not sure about the manufacturer and the equipment that we're going to use. There are 2 main ones for the terrestrial sensing, and there are pros and cons. The one company is more robust, hardier for the Arctic environment, but they also don't have the wireless capacities. And because we're going to be putting out a network of a lot, we're thinking 20 to 30 of these around the city. The wireless is really important for us. So, but still haven't made the final decision.
Are you guys designing the nodes yourself, or are you buying them from a third party, or having them built by a third party?
So we're going to do a mix. We're going to buy a bunch. We're going to buy some pretty robust setups from either Campbell or Onset, and then with Philippe Rosado Murillo, we're probably going to design some of our own sensors. Those might be some air pollution sensors, and maybe some other things, carbon dioxide, we haven't decided yet what we're going to use for snow. We're actually thinking of maybe using citizen science for snow. I guess that's a sensor in and of itself. But basically, local residents can go out and, poke a meter stick or whatever into the snow and measure the depth at certain frequencies.
Awesome. Other questions from other folks?
I think I botched the false positive and false negative. I don't know. My brain just sort of clicked into a different gear. I'm guessing I'm guessing what a false positive, false negative is, but I don't think I did it correctly. Maybe I did, but
oh, that's no problem. I think you corrected it after the fact.
Yeah.
Looks like we've got one in the chat. I'll read it out loud. It's from John Laurie, who says, "How did you establish ground truth for the drone project? And did you see drift in criteria among locations and light conditions?"
So, so ground truthing was Kelsey basically going out in the field and mapping, mapping individuals in both the training and the testing data set, with pretty robust GPS that gets you pretty close with regard to accuracy. So that was the ground truthing there. And the drift with regard to light conditions.
Yeah, locations and light conditions.
Yeah, that's challenging. So what Kelsey did was she would only fly the drone under at certain times of the day. So like the I think 10 to 2, so midday period. She would only fly it under pretty static atmospheric conditions, meaning not variable clouds. So your light environment isn't changing really dramatically over the course of your flights. There's calibration. So there's a pure white pad that you put out, and well, there's black as well, but you can do that just by closing the shutter. But so there's a white and a dark calibration. So what you're getting is, so even if your conditions are changing, what you're getting from flight to flight is relative in a way. It's relative to your references. So that helps with that. And then the other thing was that in selecting the pixels for the analysis, she would look at the image and only select well-lit pixels. So she did a lot to ensure that to minimize any conditions that were, and to minimize any differences that were related to light conditions or other conditions.
That makes sense. Another question in the chat from Juliana. She asked, what is the price point for the hyperspectral imager you used? And what are some examples of spectral bands of interest currently outside of the current sensitivity? Oh, yeah. Okay, so good question. The drone is the cheaper of the two. The payload is the more expensive. I think, if I'm remembering correctly, that it was in the 50 to 60,000 for the camera, for the hyperspectral sensor. And the drone is, I think, is in the 5 to 10,000 range for that model. And so the shortwave infrared, there are some important water absorption features out there in the shortwave infrared, and I believe absorption of other atmospheric constituents. To be very honest, I'm really not that familiar with the shortwave because I haven't had a great opportunity to use it, because I haven't really had
I haven't really had instruments yet that take advantage of it. In this, when you, we have handheld instruments and drone-based instruments that do hyperspectral, and the price point goes way up when you add that shortwave infrared. And even with the drone, it would have been a completely different sensor. So it actually would have been 2 sensors on the same drone. I don't even know if they're, if it, well, I'm not even 100% sure the drone has the capacity for it. And then you have to coordinate 2 separate sensors, which is a pretty tricky thing to do. So anyway, I'm not super familiar with the shortwave, but it definitely, absolutely, there are water absorption features that are important out there.
Great. I think we should probably transition to our next talk. I'm sure there are lots of interesting discussions coming out of your talk, but just want to make sure we give enough time to Professor Cocroft. So thank you again, Professor Epstein. That was really set the scene and super interesting to hear about what you guys are working on at lots of different scales and lots of different places. Awesome. So we'll transition to our second talk. Next up, we have Professor Rex Cocroft, who is a professor of biological sciences at the University of Missouri. And his research focuses on behavioral ecology and evolution, and specifically the hidden world of insect communication. His group studies the evolution of communication and its role in divergence, speciation, and social behavior among plant-feeding insects. Social and ecological interactions among insects often involve vibration, so his work specifically investigates important features of this widespread but understudied form of communication. And most recently, he's been focused on investigating plants' abilities to perceive insect-generated vibrations and respond adaptively to them. So we're thrilled to have you, Professor Cocroft, and I will turn it over to you. Thank you.
Well, thanks so much for the invitation, Joe, and thanks for the introduction, Carolyn. I will now share my screen. And I really, I find the mission of the Media Lab very appealing, in part because I was, well, especially the melding of science and art, because I was actually a piano major in college. And then when I decided to pursue what was really my first love, which was biology, that experience of being immersed in kind of analyzing sounds really, I think, led me to study animal acoustic communication. And I was drawn on the one hand by those intellectual challenges of understanding the coevolution of signalers and receivers, but I was also drawn by just the incredible aesthetic experience of studying signals that have evolved because receivers in some very distant species find them attractive. So before I get going, I'll just have a brief visual acknowledgement of folks in my lab. And as you've talked about in this course, so the mechanical vibrations that are traveling through structures carry a tremendous amount of information. And the structures that I deal with in my research are the stems and leaves of living plants. And we all know that plants need to be strong to support their weight, and they need to be flexible so they can bend with the wind or other forces, and they need to be resilient so they can restore their original shape after being deflected. And all those properties make plants really excellent structures for transmitting mechanical waves. So there are really a lot of organisms that rely on those mechanical waves traveling through plants for monitoring the environment, or they maybe introduce mechanical vibrations into plants. And when I say a lot, I really mean a lot. So you can see from the quote that this is, the interaction of plants and their associated arthropod community really is makes up a lot of species, and a lot of those ecological interactions in turn depend on behavioral interactions that are based on information transmitted through plant-borne vibrations. So let me just say a little bit, since this is a sensing course, about the kinds of waves that we find in plants. And so bending waves are well established as being kind of the predominant form of wave in plant stems and also in leaves. But that's clearly, there's a lot of room for additional work. And one of the features of bending waves, for example, is that the propagation velocity is proportional to the square root of frequency. So they're dispersive, but a recent paper from another lab showed that actually above a certain frequency, depending on the diameter of the stem, the waves are non-dispersive. And so there are clearly more things going on, and there are many kinds of plant structures where we really haven't studied exactly what's going on. So that. Okay. So what I want to do then in this talk, Zoom permitting, is talk a little bit about that vibrational soundscape of living plants. What are the sources of mechanical energy in plants? And a bit of, I'll come at it a bit from my background as an insect biologist, and then I'll talk I'll finish up talking more about our more recent directions of looking at the plant side of this. So if we can use this little idealized pea plant as our model and think about, okay, well, what are the sources of mechanical waves in this plant? Well, there are off-plant sources, such as wind or acoustic sources in the environment. We know leaves are very good absorbers of acoustic energy, rain, soil-borne insects, flying insects. And then there's a whole suite of on-plant sources of acoustic energy, including insects walking or moving on the plant, insects singing on the plant, insects feeding on the plant, or insects pollinating the plant. And in all, you can get a very complicated soundscape with many different components. And you know, here's, you don't think of maybe this is part of the vibrational soundscape of a plant. You can hear cardinals and robins, but that was recorded just from the leaf of a plant. Here's one of those on-plant sources, one of many species that signal through plants. Can you hear that? Yeah, okay. I love this because you can actually hear the beat frequencies there. And so what you can get is a I'm just going to switch to the Zoom screen just so you can hear this. So you don't really need to see this. This is the acoustic, the vibrational environment recorded on a single grass blade. And those of you who know birds might hear a Carolina wren, you might hear cicadas, and then in the foreground you hear leafhoppers. So all that is the environment of a single grass blade. And so what we really are still lacking of a broad scale view of what natural vibrational soundscapes are like, but clearly they're highly local because plant species attract different insects, different plant parts attract different insects, and there will be those off-plant sources that may be shared, but even then the plants may respond to them differently. These tend to be rather transient because insects may move through the environment or only be active for a part of the day. And they have a lot of information about the activity of the insects on those plants. So, you know, this being a class on sensors, really you should have spent your, that was a wonderful talk. I won't say, you should have spent your semester studying insects. It's totally tongue in cheek, but insects are just walking, flying, burrowing sensor arrays. They're sensing light, they're sensing temperature, they're sensing humidity, they're sensing electric fields, they're sensing sound, touch, vibration, airflow. And when it comes to vibration, they do have structures of various parts of their body, but the most important for the insects that I work with, and for most insects, are in the legs. And so here's a diagram of the leg of a honeybee, and the dark line running down through the center is the leg nerve. And then you can see at each joint there are these little sensor bodies, and those are all mechanically gated, stretch activated receptors, most of them for sensing the insect's, the position of its own limbs, but there's one called the subgenual organ below the knee that is really important for higher frequency vibrations. And in honeybees, actually, when they're really young, you can see through the tibia and see the subgenual organ. And so if you look at this photograph, if you look at that little red arrow on the left, you'll see that there's some dots, and that's, you can see the position of the subgenual organ when the leg is moving down, and then the position of the subgenual organ when the leg is moving up. And the difference is subtle, but essentially that organ, it's stretch activated, and it's based on inertial lag between the fluid that it's embedded in inside the leg and the exoskeleton of the leg itself. And Joe asked me if I'd talk a little bit about the sensors that we use, and I decided to refuse to do that because I wanted to pose you the challenge. This is a course on sensors, and so I'll ask you, you can email me later, you can tell me right now, you can tell me later, where do you think the field should be going? Right now, the field is based on piezo accelerometers and other piezo sensors, on laser vibrometers. And this has been one of my perennial disappointments. And there are neat things going on, like the famous visual microphone from MIT, or some neat biomimetic sensors being developed. But those are really not in day-to-day use yet. So, well, what are some of the kinds of things insects are sensing? Well, here's one of the insects related to some of the ones that I species. It's a little that I study. It's a little 4-centimeter insect that lives on oak trees. And that's the kind of signal that insects are introducing into plants to communicate. This is a chorusing tree hopper. Well, you know what I'll do is I'll just let you hear the sounds by animating it. Not very insect-like for what you're probably thinking about.
We can't hear that, Rex, actually. We heard it before, but when you
Okay.
go review, we don't hear it.
Okay. All right. Well, so in any case, there are all these insect-created vibrations, all these mechanical waves traveling through the plant. And keep in mind that they're traveling through a living organism. And so it's amazing how long it took me to really think about this. But several years ago, I met someone who is now a long-term collaborator, Dr. Heidi Apple, and I had recently written a review of how insects use plant-borne vibrations. And she introduced herself as studying how plants recognize that they've been attacked by herbivores and defend against them. And in that conversation, we sort of both thought at the same time, well, there's all this information about the herbivore community traveling through the plant. Could the plant be making use of any of this? And I'll just build, before I present data, the sort of thinking behind why we thought this might be possible. Of course, we really didn't think it would be going on. But if you think about plants and mechanosensing, you probably think first of like really specialized structures like the Venus flytrap, where there's trigger hairs and an insect walks past at the right rhythm, and then the trap closes. But that's really not what you should be thinking about, because plants in general have mechanosensory ion channels in virtually every cell. A lot of these are for sensing turgor pressure and sensing other kinds of stresses. Some of them are involved in sensing vibration, and we don't yet know in plants very much about which ones do what. But some of them are terms you'd recognize if you're familiar with ion channels in humans or in other organisms like the piezo channels. And these are tension gated. They're situated in membranes, and they're virtually in every cell of the plant. And so you can be pretty sure if there's a mechanical disturbance in a plant, unless it's super high frequency, the plant can probably perceive it. And on the other hand, you're also probably aware that there is this history of people playing sounds to plants. And an aspect of this literature you might not be aware of is in Asia, people have used this for years to place sound to crops. And they'll take a single frequency and they'll do experiments to figure out which frequency to use and actually increase the yield of some crops by playing them sound of a particular frequency. And here's an example of another a kind of stimulus that's been used a lot. This is just a kind of a random study of how algal growth was influenced by sounds, and they played two different songs, right? And so the blues song you can see caused greater growth. And what drives me crazy about all of these studies is that there's no relationship at all to plant ecology. Why on earth would a 300 hertz tone cause a field full of cotton plants to change their investment in growth versus reproduction versus defense? And this field really doesn't consider that. And playing music to plants, as much as I identify with playing music, but it's kind of a fool's errand, because music is a huge combinatorial library of so many frequencies and rhythms and amplitudes that you really have no idea what you're doing. And so Heidi and I are both more ecology based, and so what made sense to us was to ask, what are the sources of acoustic or vibrational input to plants in their natural populations, and then what kind of response to those stimuli would be meaningful. And the first thing that came to my mind is these creatures. Now, are you able to hear that? After studying insect signals, initially I found this to be an unpleasant sound, and now I find it very pleasant. It's like sheep grazing in a meadow. Lots of things have to be going right to be able to hear this. But I knew from having done field work on signals, if they're feeding caterpillars on the plant, you're gonna either need to find a different plant or find them and move them off the plant, because those are high amplitude, very repetitive signals that are much higher amplitude than most of the insect signals that I was trying to record. And so there's some rationale for thinking that maybe plants should be able to detect signals. And I figured, and Heidi agreed, if there's any signal that should be interesting to a plant, it's that sound of feeding. The insect is actually cutting through the plant's own tissues. So that's real time. And so let's think just a minute about what happens when an insect bites a plant. And so there's a perception stage. So the plant needs to perceive that this is a herbivore attack as opposed to some other kind of damage. Then this initiates a really amazing set of signals, from electrical signals to reactive oxygen species to calcium signals, various plant hormones, like really a very active set of signals that then cause changes in gene expression downstream that lead to the production of chemical defenses, of toxins that can slow the growth of the caterpillar, deter it from eating, or actually poison it. And the other kind of defenses that they use are these airborne volatiles, which don't necessarily have a direct effect on the herbivore, but can call in its natural enemies. And so that can happen, or after the plant perceives a herbivore cue, it can become primed, so that you don't see an increase in chemicals right away, but you see that when you do challenge it with herbivory, it responds more strongly or more quickly. And so we wanted to ask, well, okay, could feeding vibrations play a role in the initial plant perception of an attack? And we know the plants can perceive those damaged products, damaged cells, things that are outside the cell that shouldn't be there. There's some really important compounds in insect saliva that plants have evolved the ability to detect. And I did sort of think, well, why be redundant and use this other cue when they have some very good cues? But we decided we would try it anyway. So we got out a trusty laser vibrometer, one of the vibrometers from my lab, and we recorded the leaf vibrations that occur when a caterpillar is feeding on a leaf. And we used the lab rat plant, Arabidopsis thaliana, little plants that really are not very easily studied in the field, but are great for the lab. And then we, since I'd worked with bug signals and trying to reproduce insect signals, it was very easy for me to then reproduce those vibrations to a new plant that had never experienced a caterpillar. And I know the hardware and the signal processing and the software to be able to really reproduce those vibrations very, very closely. Here's kind of a random example showing that, well, here's the original recording, here's the playback recording, and there's the spectra of the two. And we play these various combinations of stimuli back to multiple plants at a time. And so what we initially did in our first experiment was test for priming. If the plants experience these feeding vibrations for a couple of hours, and then you challenge them with herbivory, will they respond more strongly? And we gave them 48 hours to really build up those chemical responses. And here the y-axis is one of the very characteristic toxins that's present in mustard plants. It's actually what gives mustard its flavor, these mustard oils that can be very unpleasant for insects. And as a function of the control treatment versus the plants that have been exposed to feeding vibrations. And so in both the leaf that we played back to and a leaf on the opposite side of the plant, we saw an increase once the plants were challenged with herbivory. They seemed to really be reacting to these feeding vibrations. Not only that, but the higher the amplitude of those vibrations, the stronger the chemical response. This was just still our first experiment, and I thought, yeah, I don't know. You know, are they specific at all? And so here's another sort of bioassay of defense on the y-axis. And then here we looked at, well, the control versus chewing vibrations versus leafhopper song versus wind, and the plants only increased defenses in response to those chewing vibrations, even though the leafhopper song had a very similar spectra. And so we, after this, I was convinced enough that, okay, let's publish this, and that, you know, the public reaction was, it came out in July, like a really low point in the news cycle, and so there was a very amusing level of public reaction. I mean, the more negative was that we actually got called wacko scientist on air by Rush Limbaugh himself. So I had to get, you know, t-shirts for the lab, wacko scientists. The playback also influenced the production of those airborne volatiles that can draw in enemies of the plant. So here's the emission rate on the y-axis of one airborne volatile that actually increased. Here the blue and the orange show plants that did not have any herbivory treatment, or the ones that did. It doesn't matter. In either case, just the vibrations alone caused the plant to increase its production of this component of its volatile blend. And I'll just end here by saying that one of the more recent things we've done is to ask, basically, is the chewing caterpillar a chewing caterpillar? Do plants that are fed on by multiple species distinguish among them? So a student in my lab used recordings of, this is our canonical species that we've used. And here's another species that's a generalist. You can hear it has a much faster rate of feeding. But if you look at the waveforms of those feeding vibrations, you can see there's some differences, but they're mostly in the rate. And if you look at the amplitude spectra, they're really very, very similar. And so to us, it would be difficult to distinguish between different species of caterpillars, certainly based on spectral cues. But the question is, well, what about the plants? And so here's, again, a bioassay of defense on the y-axis, and then on the x-axis, the line indicates control, no vibration, and then the two waveforms indicate playbacks of the two different caterpillars. And the plant treated them just the same. The response was different on younger and older leaves, but there really was no difference in the plant's response to those two caterpillars, even though their feeding rates were somewhat different. And so really, it does seem that as far as the plant's concerned, a munching caterpillar is a munching caterpillar. And I actually felt that this helped me understand maybe why plants would use these cues, because many of the chemical cues are very specific to individual species interactions, but here the plants could have one set of sensory filters that would identify a whole suite of herbivores, and then the specificity would come in other ways. But there's this very consistent, very repeatable kind of acoustic target that plants could use to perceive their herbivores. So, some current directions, we're interested in, well, how widespread is this in plants? So far, I presented you data from one plant species. We have, with a collaborator, also data from domesticated tobacco. Chewing vibrations of a little caterpillar cause the nicotine to spike beautifully. But what about other plants? We think it should be widespread. There's nothing special about Arabidopsis in terms of sensing vibration. All plants can do this. And we've got some plants that have mutant lines that are defective in one or more mechanoreceptors. And so we're starting to look at, well, can we use this to figure out which mechanoreceptors are involved in sensing these vibrations? And we want to really take it out into the field and say, well, what does this really happen in nature? What are the conditions that might permit or limit a plant's use of vibrations as a herbivory cue? So far, we've never used any sort of playback in the presence of noise. And so if it's windy, how does this influence the plant's ability to sense herbivores based on their vibrations? So I'm going to end it there, and happy to take any questions.
Fantastic. Thank you so much. It was fun to hear the sounds, and I appreciate you persevering despite the Zoom issues.
Likewise.
So yeah, again, if people either want to just speak up or throw a question in the chat, I can read them out.
Yeah, just adding my enthusiasm. This is just wonderful, Rex. Despite these minor Zoom issues, it was still great. And for those of you who want to hear more of these fascinating sounds too, Rex's website, his group has got lots of examples. It's a whole other world, and to see the meaning behind that being decoded is incredible too. I can ask maybe one quick question. You said you started with people playing tones, if not music, to plants. Music, of course, can mean lots of things to a plant, but a tone you tend to think would be something specific. Any idea why plants grow better with certain frequencies? Are you emulating a predator, or is something that they like? Or
Yeah, I mean, it's a great question, and I feel that there's a whole set of whole tradition of papers that are just there to be mined for exactly that kind of question. And it's not necessarily that the plants are growing better, it's that, right, for a given amount of light and water and nutrients, the plant can invest either in growing taller or growing more leaves or bigger leaves. It can invest in reproducing, producing more flowers, fruits, or it can invest in defending itself against its herbivores. And so something about this can cause plants to invest more in growth or reproduction rather than defense. So I don't really know what to make of that. There's another example of the a couple of other labs have started trying to use more naturalistic stimuli, and my feeling is you always need to start by putting a sensor out there in the environment. But one looked at plant roots. There's a woman named Monica Galliano, and she just did a little simple, like a Y maze experiment for plant roots that grew towards the source of a soil-borne sound. In this case, she recorded the sound of water running through a tube. But if you played noise on the other side, they didn't distinguish. We still don't know like how many plants out there. I have had sensors in the ground for other insect projects, and most plants, in my experience, don't have sources of flowing water there. So I'm not sure we really understand that, but the roots do grow towards sound. There was another study 2 years ago from Tel Aviv University where it looked at recorded the sounds of bees near evening primrose flowers, and then played that sound back through the air and measured the nectar produced, and found The title of the paper was like, bee wing buzzes increase floral nectar production and quantity within minutes. And so that's a very interesting study of an example of maybe a real world stimulus. And that study's been sort of controversial, but I think it's, I definitely think there's something to it. So what those other stimuli mean, those pure tones and things, the natural stimuli out there don't typically consist of pure tones, right? They're much more complex. So it's a great question, but I don't know the answer.
Great. There are a bunch of questions in the chat. So it looks like the first one, I think you just sort of answered. The second one from Shu Guang is, do plants have G protein coupled receptors for signaling?
So here you're hitting on my limitations as a plant biologist and as opposed to an insect biologist. And so what happens downstream of those membrane-bound channels? I don't have a good answer for you of what the next downstream steps are.
Yeah, I don't have the biology background either. I don't know if Shu Guang, do you want to follow up or respond?
That's an interesting question.
Okay, I'll move to the next one. Let's see. So this is from Lino, who asks, what understanding do you have today of the way vibrations are perceived by the plants?
So that really is an extremely key question of how plants perceive these vibrations. So we collaborate with a woman named Elizabeth Haswell at Washington University, and she looks at a couple of different families of mechanoreceptors, but in some ways with these mutant phenotypes, mutant lines, she's looking for a phenotype. The plant, it's not always clear. And so we know that some MSL proteins are involved in sensing very low frequencies from wind. And we have some evidence that we're trying to follow up on that there's a triple mutant deficient in some of the MSL family members that did not produce a response to feeding vibrations. And so we're trying to follow up that now and see if that channel family could be involved. Yeah, I would love to know that.
Great. Yeah, and anyone, if you gave the question and want to sort of chime in and respond, please feel free. We can have it be sort of an open conversation. But I'll just keep reading them out loud. So the next question is from Ron, who asked, can you please make a comment about what kind of nervous system a plant uses?
So that's another wonderful question. So plants don't have neurons. There are people that would consider themselves plant neurobiologists. It's a somewhat contentious term, but plants do have electrical signaling. They have action potentials. They have some other kinds of propagating signals. And recently they've been studied in terms of their downstream effects. So we know that those electrical signals, we don't know entirely how they're transmitted through the plant. They go from cell to cell. It might be along the vascular system in part, but they do, cause changes in gene expression downstream. So in a sense, the whole plant is connected. It's very electrically active tissue, which is why, that book, The Secret Life of Plants, which you might remember, was written back in the 50s, where they hooked a lie detector up to plants. And one of my relatives asked me one time, I was like, well, why would there be any response if you hook up a lie detector to a plant? It's like, well, okay, they do all this electrical signaling. That work, of course, it's been all debunked, but anyway, just an aside.
That's great. Okay, and next question from Rob Lewis, who asked, what was the latency of the plant's response in the anthocyanin experiments?
So that in all those cases, we sampled after after 48 hours. So we don't know, it was something less than that. But we've started collaborating with a lab that uses imaging techniques to look at rapid, early event signaling, and we're finding that signaling waves are initiated if we do like playbacks of 5 minutes, for example. And we haven't tried to go less, but signaling is initiated certainly within 5 minutes.
Okay, and then the last one here in the chat is from Kelly Heaton, who asks, have cellular mechanoreceptors been identified in life forms other than plants, indicating that other life forms may be reactive to sound?
So that's a really great question. And in fact, it seems to be a universal property of cellular life that there are mechanoreceptors, and it could be for all kinds of reasons. It might just be sensing cell turgor pressure. It might be sensing things in the environment. But yes, mechanoreceptors are really a universal across all life. It's like we're probably getting close to time here.
Maybe if we can take a few more minutes, I'll jump in with a couple of questions and maybe a comment too. Don had asked a question about artists using these sounds. And of course, that struck me too, since we do some of that at the Media Lab. But there are artists that do it. Do you know of the work of David Dunn, The Sound of the Light in the Trees?
Yeah.
It's a great piece. I don't know if you
I know
have any
David.
connection to that community, but it's fantastic to see these sounds being used.
So there's a CD released actually recently by really two composers. One is Victoria Bond, who's an sort of classical conductor and opera composer in New York, and an electric violist named Martha Mook, and David Rothenberg was also on there, and a bunch of my recordings. And they've done some really wonderful things. The CD is just called Buzz, and if anybody's interested, I'm happy to send you a link. So but I think the possibilities are really hardly tapped.
We'll track that down. David actually is an alum of the Media Lab. He graduated from Todd's group a long time ago. So there's a connection.
Oh, I didn't realize that.
Yeah, small.
Yeah, I met him several years ago.
Getting back to expanding on that a bit, let's say we do something into March, which we talked about. We got this great infrastructure, wireless, sensors all over, and we may do microenvironments. We talked a long time about instrumenting a log or a plant or something. What would you pick to instrument? What kind of a plant? We're in Plymouth, so it's seacoast, Massachusetts. It's a wetland. If you want to hear insect activity of different sorts, what kind of a target would you pick?
In terms of what sensor, or in terms of where I would put the sensor?
I think first in terms of what plant should we pick? I mean, you want to get a plant that has a lot of insect. That should be a tree, should be a log, should it be just some milkweed or whatever else?
So it's unfortunately it's a little bit difficult to predict when and where you're going to find things going on. And so for all those recordings that I have where there's lots of species signaling, I have other experiences of going out in the woods and listening to plant after plant, and maybe numbers of them not hearing anything. So I think I would say if you could, I would more pick probably environments, like if you picked rushes emerging up out of the water, let's say, and you could wire up maybe 20, then you probably have a pretty good chance. And maybe of hearing some interesting interactions. I'd put something in the water. There are aquatic bugs that make a lot of signals. And that would also, I would again, I would probably spread things out a little bit, because otherwise if you pick a single plant, you're probably, chances are there's not going to be that much going on. Maybe you'll hear the wind, or maybe you'll hear some bird song, but you might not hear a lot of interesting things. But if you could wire up multiple ones, 20, 30, well, you guys in big scale, but that I think would increase your chances a lot. And then maybe you could even have like the sensors indicating like which one is active, and then somebody could zoom in and like so you could hear the ensemble, like, okay, there's something going on, there's some singing or there's some beating, and then okay, it's coming from these sensors in this location.
This is a great project. you lamented the sensors that we have. So we can get really sensitive piezo accelerometers, which imagine one of your choices for a contact pickup on a plant. I don't know if capacitive accelerometers cut it as well. What are the problems with what you have? Laser vibrometer, of course, is expensive, big. It's a piece of lab equipment. But what are the difficulties in the accelerometers you use?
They really work very well. It's like, microphones or loudspeakers or something. Like, it's a very mature technology. They're very reliable. Um, there's an expense factor. So I'm always trying to democratize this, because these sounds are in people's backyards, and why not make them more accessible, and make research more accessible. So, just a basic, decent, miniature accelerometer, maybe 100 millivolts per G, and a signal conditioner is at maybe 800 or $1,000, which, if you're a PhD student in Latin America, might not be in your budget. So expense is one thing. And I think just also you're getting a point source. So, potentially something like the visual microphone, if and cameras are maybe lending themselves to that. I don't really know how well that would work. I haven't really tried that yet, but that might work, because you could see more of what's happening across the plant.
We know Fredo Durant and his colleagues very well that are doing that at CSAIL, and that stuff.
So I sent, so the grad student's name was Abe Davis, right? I think he's at Cornell now. And I sent him, after that article came out, it's like, okay, well, you're recording sounds that are induced in plants from outside, but there are all these native vibrations produced within the plants. And I sent him some cool signals. I didn't hear back.
He was actually in my class, my musical instruments class, a few years ago, and
Interesting.
he project based around that. But yeah, I'll try to contact him. Maybe we can do something. It's intriguing. It's a challenge, though, I'm sure, because your bag of chips is not moving at all, and that can get micro vibration. Very expensive camera. This is a plant that's in the wild, different lighting, moving on its own quite a bit. You're trying to look at different kind of motion on the plant itself. So it's a dynamic range issue. But there are signal processing techniques that can start to attack it, maybe. I think the camera's still going to be something special, not a cheap camera. But you can get arbitrarily close as well. You need a fast frame rate as well. Maybe you can do it with.
Yeah, you need it.
Yeah, go ahead.
You need a decent amount of light.
Yeah.
And a pretty decent frame rate, maybe 10 or 20K.
Yeah.
And then you need a certain amount of resolution.
Yeah. I mean, optical proximity sensors would work, because you can get microns of resolution out of them and get it pretty fast, and they're not expensive. But you'd have to put that pretty close to the plant. But a bunch of cheap sensors you could just put at points is another possibility, and they're not like a laboratory grade piezo accelerometer. Now, a capacitive accelerometer, could that work at all for this, or you just don't have the sensitivity? You have to go with piezo for what you're doing. I've never tried. I'm always willing to try anything. I mean, there's a pretty broad frequency range of signals from down in the tens of hertz to 10,000 hertz or potentially more, often less. The signals are pretty low amplitude, in terms of displacement. We're talking about a few microns, or in terms of, a velocity signal, it's down in a few millimeters per second. Yep.
So we need something pretty sensitive.
Yeah, we have close contact with the manufacturers of capacitive accelerometers, which are cheap because they have mass market behind them, and they just use MEMS. And they're getting more sensitive. So maybe. Let's see. But the piezo.
Yeah, well, I would definitely. I'm always interested in trying things, and then you probably know Ron Miles and his group at Binghamton, and I actually spent a postdoc year there. But I looked at some of the stuff that they've done with these filaments, sort of like spider silk or insect hairs that coat them with gold and put them in a magnetic field, and you have all these directional microphones, and that I don't see why you couldn't do that for accelerometers too.
We're going to have him come to give a talk in our acoustics session, but we've already got. I let the students basically populate that one out, but we'll bring that into our dialogue too. I think that's fantastic work. It's amazing. So I could keep on talking, but I think best to have you up here at the Media Lab physically at some point in the not too distant future, if you're interested. We'd love to have you hang on with a
Well,
group.
that'd be,
Give us a
that,
proper talk.
yeah, I'd love to.
Same with you, Howie. Yeah, you and I be talking at your NSF initiative unrolls too.
That'd be great.
I don't mean to take over. We're starting to collapse down to the bare bones of the class, but does anybody else have a question? Maybe then we'll let the speakers and everybody else go and just relegate it to the class and the TAs and go from there. So thanks so much, Rex and Howie. Great to see you guys.
All right.
Thank you all.
Thanks.
Thank you so much.
Good to meet you, Howie. I really enjoyed that.
Bye. Enjoyed it very much. Yeah, thank you.